and welcome back to the Computational Genomics Lab. Today we are going to analyze all of our data. Last time we compared the 0 versus 12-hour time point and saw how to get the results out of this DDS, DESEQ data structure object. And then today we're going to learn a for loop to go in and just do that automatically for all the different comparisons. So let's get started, and we're going to go over a lot of the same things we have in the previous classes just to get learn through iteration. For example, today I'm just going to load in the counts matrix from raw right out of the NF core RNAseq pipeline, and remember it's going to have that gene name in there. But before we take that out, let's make a gene ID to gene symbol that we'll want for our results tables. And we've gone over this before, so we're just going to grab the first column and the row names of the count matrix, and we'll have our G2S. It's not viewing for me. Okay, well, you've seen this before, and then we will, oh, there it is. So we have the gene ID and gene name, and then in our counts matrix, we're going to take out that gene ID column now, so with the all rows and -1 through the indexing. You could also do it this way, where you pipe in counts matrix and use the select function to remove the gene name column, and you don't have to put it in quotes because you've used tidyverse to put the whole matrix into memory so it knows the columns names. Okay, then we round our data, as right now it's got decimal places in our counts matrix. Again, stuff we've all done in previous videos, just to go through it quickly here, and we're going to load our sample sheet, which we just did, and then we need to factor our sample sheet. Let's take a look at that one actually real quick. So what we're going to do is make our, oh, it's already factored actually, so we saved it factored. That's great. You can always factor it again. It doesn't really matter. So we do see here that the sample sheet we loaded in from the previous class has already been factored. Good way to save it. So you don't have to do it again, but sometimes it's just good to do that. So now the time point and replicate can be read in by DESEQ as a comparison. And here are our unique sample IDs, which are going to match the column names of our counts integers, what we've called it after we rounded. So these column names are the same as these row names, and that's going to be an important check that you're going to want to always do. Why not factor again? As I mentioned, it's pretty important. DESEQ requires it. And so now we're going to do a little check here. We're going to say, okay, if all the call names in counts integer, so that's the column sample names, aren't directly equal to the row names in the sample ID column, caching using the cache sign to index into our sample sheet into the sample ID column, stop if this is not true. And that's very important, because if there was a mislabeling or one column got moved around, the analysis would be completely off. So let's add this checkpoint here. Good, didn't stop, so everything's in order. And we're going to run our DESEQ. I'm going to start it just because it tends to take a second. And again, we're using this DESEQ data set from matrix function, same as we've been doing it. Our counts data equals our counts integer, which has the rounded values. Our call data is going to be our DESEQ samples to tell it what samples to compare. And the design here is going to be just time point for now, and as we get into the class we might do some different statistical tests. Okay, our DESEQ has run, and we can check our result names using the result names function. And we called it this time DDS time point. It's a little different than last time. It doesn't really matter, and it didn't come up in my console down here because what I did is I made results name an object, and that's going to be important for our for loop. That if we look at this in here, we have our intercept, our 12 versus 0, 24 versus 0, 48, 96. Okay, so now we could do a for loop saying for each row in here, do that results, grab the results with the results function, and do that iteratively and merge it all together into one table. That's what we're about to do. We want to get rid of this intercept here because it is not does not contain the data. So I'm going to do results names minus the first row, and I could put comma here, all columns. It doesn't really matter. I'm just going to take out that intercept, and if we view it, we can see now it's just our time points or result names we want to go grab. And last time we just fed in this with that contrast function. And when you're doing this in the future, you will probably always use a for loop or an lapply or sapply, which we'll talk about in a second. Okay, so first things first, we're going to make a generic data frame that's just going to have column names here. So we'll call it ResDF, and we'll use our data.frame function to start like making an open blank Excel sheet. Make a column called gene ID, the base mean. These are all the results in that DESEQ results file we looked at in the last class. So essentially we're just setting this up so when the for loop runs, it'll populate this table with each of these columns. Okay, so now if we look at this though, it's just empty and blank, no data. So let's fill it up with a for loop. And again, in here, I'm going to go through this step by step, written out much more clearly. I'm going to go a little faster in the videos, and then you can always go back and read more about what's going on here. Okay, so for loop is important. You're going to use them a lot. It's also could be done with lapply or sapply. Those are R functions that are essentially built-in for loops. They're going to say, for each row, I'm going to do this function. Super handy. But we'll do just a classic for loop nomenclature for now. So we're going to say for, and then in parentheses is what we're going to operate on. I in 1 through length results names. And if we remember, results names are our different time point comparisons. So it's going to have I as the first row. It's going to go through, grab out the results, and pipe it into that data frame. And then it's going to go to the next one, until there's no more rows left in our results names function. Okay, so we are going to take results names and results names I. So we're grabbing the time point information, essentially, that 12 versus 0 time point. We're going to keep track of it as it goes through the for loop to ensure it doesn't write over the other time points when it's looping. And then we're going to make a res object here. And this is the results function we used last time that's going to give us all those information that we really want. That's like the key function here is results. For the DDS time point is the DDS we just made. And the name equals results name. Again, just keeping track of that name. And then this is where it kind of gets confusing. Inside the for loop, we're going to make a little mini data frame and call it temporary ResDF. And we're going to put in there res, which we just got all the results from our DESEQ object in res. So res is going to come in. It's going to turn into a data frame here. It's going to be stored as this object. And then we're going to do row names to columns. So we're going to take out those row names from metadata into an actual column. And we're going to merge it with our G2S. And we had to do that so that they can find keys that match. So G2S has gene ID in it. And right now our gene ID is in meta. So we're going to put it into a column. Those two are going to match and bring in the gene name. And then mutate is something that's also very powerful in R. It makes a new column and you can run a function like you can multiply everything times 2 and put it in a new column. So you give it the column name and we're going to change result name to results name. There's an extra S. So this is just cleaning up the name of that column. And then we're going to take that ResDF, which is empty, we just made, 0 observations here. And we're going to do bind rows with ResDF. So after this first iteration, we're going to have the 0 versus 12 hours in ResDF, all the data for every gene. And then we're going to bind it with our temporary ResDF that we just made above. So the first round, it won't really need to do anything. It'll just populate that thing. But on the second time point, it's going to take this data frame of 0 versus 12, and it's going to make 0 versus 24, and then 0 versus 48. It's going to like stack them through this bind rows function. Just think of them as 0 versus 12, 24, 48, 96. And we'll recorded the time point in a column. And that's it. That will do it for 1 sample, 100 samples. So you can just use this if you want. I challenge you to try and use sapply or lapply to do the same thing. It would work. And a little more for loop. So we have this for loop here in our results name, and the for loop's going to do everything inside these curly brackets, so we have to have them ended here to stop the for loop from running. Okay, so let's run that. And it's going and it's thinking. It's going to take a while because it's grabbing all that data. Wow, okay. So now we can see in our, it also told us how many eyes there were and kept these result names as values in here. But we can also see, oops, I'm going to set as my working directory like I always say to do. Our ResDF has 221,000 observations. Okay, cool. So we're going to see some NAs in here. Again, we've looked at those before. And so what was important is we kept that result name in the for loop. You can go back and look at where that came in. But that way, we didn't just rewrite over each time point as we were loading it. So we can see here that we also have the 96, which is great. And we could scroll around, make sure we have the 48, but I'm sure we do. And we have all of these values, including the gene name. Super handy. Took a second, and we just took 4 different comparisons and made it into one big table. And then that way we can kind of filter and find our significant genes. Okay, so let's check our NAs. We saw that there were some in there. And it's kind of a bummer because they're character and it will mess up some downstream analyses. But we're going to leave them in there for now and just move forward. But it's always just good to remember how much of my data is actually not there. And so let's take a look at that. So is.na is going to look, and if it's true, it's a logical statement that if there's an NA, it's going to say, oh, that's true. And then we're going to take the sum of how many times that was true. And we're going to look in our p-adjusted column, which is our significance call, an adjusted p-value. That we're going to use a cutoff of 0.05. And we're not going to use the p-value. The p-adjusted is important because it takes a distribution of p-values. And it says, hey, look, I see this many across all p-values, so I'm going to drop those and have a little more stringent value for my p-value. Okay, so let's see how many there are. Holy cow, 157,000. That's a lot. Okay, so that basically means DESEQ had trouble with the base mean or some aspect of the data. It was like, you know, I don't have a good measure on this gene, but that's okay. And I just wanted to point that out because this happens a lot, that some of your data just isn't measured in RNAseq. There's a lot of genes and they're not in every cell, and that's just the reality. So let's save our ResDF. That's all of our results. That's all we need for the rest of the class right there for this, at least this data set. And we're going to save it in our results folder, and we're going to call it time point ResDF. So super handy. Now we have all of our data in one table, and so we can save that into our results folder as an r.data. And same thing here. We gave it the object and the file name. The object is our DDS. You don't really need to save these. You saw how fast you can make them, but for now we'll do it just for the sake of saving. Voila, we have it. Okay, so I have some more information on p-adjusted, why we're going to use the p-adjusted here, and a web link to more information on that, and a little exercise to go in and explore your data. What gene was the most significant? Who had the biggest fold change? What time point had more genes? That would be pretty tricky. But you have all the data you need to do your analyses now. And in the next lesson, we're going to go look around and do some of this and filter and find out what DOX is doing to stem cells.